Hello everybody, welcome once again to the Vitrolife Group Academy studio here at ESHRE in Amsterdam. Our next session, we have two esteemed gentlemen here, Nuno Costa-Borges and Mark Larman. We are going to talk now about the safety of our culture media. How do we test our culture media? How do we know that the culture media that we're using is not going to affect our patients? Gentlemen, over to you.
Thank you, Jaco, and thank you, Nuno, for joining us today in the studio. Obviously, the quality of your media and I guess any device within the IVF lab is going to be determined by the quality of your Mouse Embryo Assay testing. And you run an independent lab that runs Mouse Embryo Assay. So maybe you could start by introducing yourself and Embryotools.
Okay, thank you. Thank you so much for the invitation. I'm very glad to be here. Just to provide some context, I'm a scientific director of Embryotools and also co-founder. Embryotools is a private company that is established in Barcelona at the Scientific Park. And essentially, we have a quality control testing center. We also offer IVF training and consulting services. And our laboratory is accredited. And within the scope of our accreditation, we include all the quality control tests that we perform, the mouse embryo assays, sperm survival assay, and the endotoxin assay. And among these, the MIA test is the most requested. We roughly perform 3,000 MIA tests per year.
Okay, so that's pretty similar to the number of MIA tests we run in our Denver facility. So very similar. And in terms of the actual Mouse Embryo Assay, obviously it's an embryo toxicity assay, but how would you define it further?
Yeah, I think the primary aim of a mouse embryo assay is, as you mentioned, detecting the toxicity or that some products are suboptimal before they enter in contact with the human samples. Mm-hmm. And this, I think, is the way we can define a MIA test. But also, I think it's very important that the MIA test is sensitive and that we optimize it carefully, not just to test the toxicity, but also the functionality of this product. And I have in mind, for example, vitrification kits. If we expose mouse embryos sequentially to the vitrification and warming solutions without actually vitrifying them, we will be still doing a MIA test, but we will not be testing the functionality, so that in case of a critical error in production, for example, forgetting adding a cryopreserver in the solution, would go undetected, and the product would have passed the MIA test.
So it's not just a toxicity assay; it can be used as a functional assay too.
Exactly. I think I like this definition. I think is more appropriate.
Mm-hmm.
Yeah.
As a medical device supplier, we obviously have to perform a mouse embryo assay. That's dictated by the regulatory notified bodies. Mm-hmm. There hasn't been really much guidance until the FDA released their guideline on how to perform a mouse embryo assay. Mm-hmm. So we do have at least some guidance from at least one regulatory body. How do you view it from an industry perspective?
Yeah, you're right. So I think the majority of regulatory bodies follow this document from FDA. It's actually a guideline that somehow establish this acceptance criteria that we have accepted as a rule of 80% expanded blastocyst formation rates. But it's just a generic document and also non-binding. And this opens opportunity for testing companies or manufacturers to test their products on their own way. Mm-hmm. And therefore there's somehow lack of standardization of these assays, and some can be more rigorous than others, and that's where we noticed differences in quality of the products.
Right. So there's no ISO standard for the mouse embryo assay?
No, I don't think. Mm-hmm. Yeah.
So I guess that leads us on to the next question. So why is it important to understand how a mouse embryo assay is performed?
Yeah, we have examples of situations that happen in the history of IVF, and also recently episodes that products that were tested with mouse embryos were launched into the market, and then they have an impact in the clinical outcomes. And this means that there might be differences in the rigor or the sensitivity of how these assays are being conducted, because if they are tested, it was supposed that these products shouldn't have become available to the market.
Right.
So actually, you have I think an interesting talk this afternoon from our lab manager, Dr. Enrique Mestres, where we had the opportunity to test some products that were on the market, and they had a very negative effect in the clinical outcomes and were devastating for patients. And we had the chance to test them using our protocols, and the results were very interesting, and we're presenting here this afternoon.
I guess that's one of the challenges when you don't have any ISO standard or strict guidelines, right, on how to perform the mass embryo assay. So we spoke previously, and we kind of came up with a list of some of the factors that we think impact the sensitivity, and we'll discuss some of those today. But we actually do have a webinar on our website where we had our MIA lab manager, Eric.
I've seen it.
You've seen it, right?
So
we went into a little bit more detail in that webinar and provide some examples of why we think these factors increase the sensitivity. But maybe we could go through this list now. The first one being the strain of mouse. At Vitrolife, we use the F1 strain to test all of our finished products. We do use an outbred strain to test some of our critical raw materials like albumin. But what are your thoughts around different strains of mice?
Yeah, there's some work done on this. It's clear that there's a difference between hybrid strains and outbred, for example. What we know at the moment is that hybrid strains, when we provide good conditions for them to develop, for the embryos from this strain to develop, we have consistently good results, which I think is good, because it allows us to establish what we should expect from this as a result from this culture of these hybrid embryos. And using the same conditions, but culturing outbred embryos, we know that at most we can have about 60%, which means there's a clear difference in sensitivity between the two different
right. So you couldn't use
strains.
those with the current acceptance criteria of 80%, right?
So, but I think it's a good approach, the one you're following, because if for raw materials, particularly those that are more critical, like you said, the albumin, for example, if you have the chance of testing it with this sensitive strain before adding to the final product, I think it's a very smart approach, and means a lot about how Vitrolife, I think, cares about the quality of their products, right?
Yeah, and then there's been some discussion around using the bovine as a model.
Yeah.
What do you think about using bovine embryos instead, or in addition to mouse embryos?
Yeah. I'm aware that there's been a discussion on this as well. In my opinion, the bovine model, as well as other animal models, for example, the rabbits, are interesting models for testing products that are still in development, for example, to test their suitability before doing translational work to humans. And what we usually call proof of concept in different species, to demonstrate that the product is okay to go to test in humans. However, I believe there's some challenges that we need to consider if we want to apply these bovine embryo assays as a routine for testing. I'm thinking, for example, that the original material for this test would be the slaughterhouse ovaries. That to collect these oocytes is challenging. We need to in vitro mature them. We will need to inseminate this in vitro. We need enough number of eggs to start with to do all the procedures. It's a lot of work, costs. On top of that, we would need to demonstrate the traceability to comply with ISO regulations. So I think to implement this and consider this as a possible option to substitute the current MIA tests, it would be different. And from what I know, there's not enough studies also, or either to demonstrate that the bovine embryo should be more sensitive than a mere test performed with rigor or
Exactly.
optimized for testing toxicity.
Yeah.
So
Yeah, we've done some internal testing ourselves, and it does suggest that if you perform a sensitive mouse embryo assay, it's actually more sensitive than using bovine embryos. What about number of cells? So we always use a 1-cell mouse embryo assay for all of our tests, but the FDA guidance does stipulate you can use a 1 or a 2-cell.
yeah, that's something that that we also, I don't think it's so so interesting to mention. I'm not sure if FDA should mention this on the guidelines, because we have enough evidence that one cell stage embryos are much more sensitive than the 2-cell, particularly because in the mouse species we know that the genome activates at the 2-cell stage. So this makes the embryo already more robust than if we start with 24 hours earlier, right? Apart of that, the duration of the assay would be shortened in 24 hours, so the product would be in direct contact with the mouse embryos. We're losing 24 hours of exposure to them.
And then culture setup. So at Vitrolife, I think our philosophy is always, we're trying to find something that's suboptimal. So when we design our culture setup, we want to stress the embryos a little bit, so if anything additionally suboptimal is introduced, then it's probably a lot more likely that we'll detect it. So for example, we use high oxygen incubators. We use a simple medium.
Yeah, this is tricky, because on one hand we need to provide what the embryos need to develop, but at the same time we cannot promote their development. So they need to reach this balance in the protocol, so that if there's something suboptimal, we can detect it. And apart of that, it's very important the environmental conditions where these MEA tests are performed, because if in case of failures, we need to be sure that the problem is associated to the product that you're testing and not from external cause.
Yeah.
So yeah, I think this is
and then
we need to play with all these variables. Like the culture medium should not be the formulation should be simple, not to promote the development. The number of embryos that you put in culture together in the droplet. Yeah, all these factors can, in our views and from the research we've been doing, I think it's a matter of how you define, you fine-tune all these parameters to make the MEA test more sensitive.
And then the assessment criteria and endpoints that you're going to measure, I think, are extremely important. We mentioned at the beginning that really it's only one that's stipulated, right, which is 80% expanded blastocysts at 96 hours. At Vitrolife, we actually monitor the embryos at different time points along embryo development, so we want to make sure that the embryos are developing at the same rate as the control. And then we actually take the blastocysts, stain them so that we can see the nuclei, and then be able to count the cells within each blastocyst, so we can then compare that to a control so that we show that it's not statistically lower than the control.
It's a very good approach as well, and I guess you do it in all your MEA tests.
Exactly,
right?
yeah, all finished products, yeah.
I think it's very important, not just the 80% blastocyst formation rates as it's established by FDA guidelines. In our experience, we follow the embryo development daily, and we noticed in some cases that we can already detect that something goes wrong in earlier stages. The embryo development starts getting slower, and in some cases, still the embryos can make expanded blastocysts and fulfill the 80% acceptance criteria. Yeah. So in the end, we can have expanded blastocysts with few cells, and we would only detect it in an objective way if we perform the cell counts and then compare the average number against the control. So I think the approach is the most accurate and objective that you can do, and we also in Embryotools we also offer this possibility. Yes.
And then I guess another important thing is actually how you set up the test. So I mean, I guess a good example would be when we test our oil. We have a couple of different types of tests, and one of them is what we consider a microdrop test, where we culture embryos individually, right, which creates a stress because you're removing the paracrine factors from other embryos. And we have it in a very small volume so that you increase the surface area to that volume. So you should be concentrating anything that is suboptimal into that media drop. So do you have any other kind of thoughts around test setup?
Yeah, the test setup is super important. Oil is a critical product that we use that stays in contact with the embryos for long. And it's we have seen episodes with oils that were extremely toxic. So the most sensitive assay you can make for testing oils, I think the best. And in our case, we have also some publications that we made comparing oils. And you notice that the presence of protein, for example, in this case, for detecting peroxides, for example, is super important. While if we had used a medium without protein, this
you
probably wouldn't see it. But on the other, on the opposite, for example, for testing plastics, we know that the protein in a medium can mask also toxicity. So in the end, we have to optimize the MIA test and the sensitivity for each type of product, and play with the different parameters that we just talked to make the test sensitive.
Great. Well, I think we've talked about a lot of different factors that can change the sensitivity. So I think a lot of this work is being performed by the teams at Embryotools and the team in Denver. So we definitely would like to thank all of those team members because they're helping to ensure that the products that we supply that are going out to the customers are of the highest quality. We're ensuring that we're testing them in a proper way.
We both have a very dedicated team, and we wouldn't be able to explain all this if it wasn't with their help.
Yeah. So definitely like to thank both teams very much. So I guess we'll go for some questions. But we did create a slide here. If you are a customer and you want to ask your supplier about the Mouse Embryo Assay, these are definitely some of the questions that you can approach your supplier and ask them how they're performing their Mouse Embryo Assay. So thank you for your attention.
Thank you.
Thank you very much, Nuno and Mark. Thank you. We'll see you at the next session.